Are you trying to ace your A&P final exam? You are in the right place. In this video, I'm going to train you to go through questions I know your professors are going to ask you and reason your way through to the right answer. And if you stick around, there's going to be a free giveaway opportunity that you can participate in. So be sure to watch the whole thing. Hey everybody, Organized Biology here, where we make difficult biology concepts simple. And if you'd like a work along with this test, you can go ahead and see the description pinned comment for a copy of the test with the right answers. But I'm going to actually go through this video, have the questions here, and reason my way, if I were taking this test, through the question itself to get to the right answer so you can be trained when you take your test. We're gonna get right into it. So the first question is, what level of structural organization is the most complex? So we're talking about the levels of organization here. It says chemical, cellular, organ, or tissue. Now, if we were to make that hierarchy, we know that we are an organism, okay? But organisms are consistent of many organ systems, right? The multiple organ systems of your body, like your cardiovascular system, and these are comprised of specific organs. So we know that this is a pretty complex tissue, so therefore organs would be the right answer. Now we know that organs are made of tissues, tissues are made from cells, and cells are made from a variety of organelles, and then macromolecules or molecules, and then atoms. So if you know the levels of organization like this, you can answer any question about it on your final exam, which I guarantee you will be there. Now the second question is, homeostasis refers to the body's ability to do what? Now if you don't know the right answer on this one, I don't know if I can help you. Homeostasis always refers to keeping your internal environment stable. So if you read those answers, change anatomical structure permanently, that doesn't really make sense. Maintain a stable internal environment, absolutely. It's going to be that B. The other two don't really make sense with homeostasis, okay? Now the third question you definitely need to know, which plane divides the body into equal right and left halves? So we need to know the planes of the body. So the first one says coronal, which is called the frontal plane. Corona actually means crown. So it's as if you're crowning yourself and you're dividing into front and back. So that's the coronal plane. Now, is that what we're looking for? No, because we're looking at right and left halves. The second one says sagittal, which is the correct answer because sagittal divides right down the middle and separates out right and left. Now the other two, the transverse will be basically a horizontal plane separating superior and inferior, and then the oblique is going to be some sort of diagonal cut of a plane. So it would have been B, sagittal. Very good. Now number 4, here's a good one. A negative feedback mechanism typically does what? Amplifies the original stimulus, reverses a change to maintain stability, produces rapid escalating responses, or is the same as positive feedback. We always draw in my class, we have some sort of homeostatic variable. Let's say like it's body temperature. And for you people in every other place of the world, our body temperature is typically 37 degrees Celsius or about, give or take, 98 degrees Fahrenheit. Now, typically, you can get too warm sometimes. So maybe you raise up to maybe 100 degrees Fahrenheit, or maybe like a 39 degrees Celsius. So you're too warm. I guess it'd be 38. Now, in that case, we are now out of homeostasis because this is our homeostatic level. So we want to oppose the change. We want to have a response where we start sweating to bring our temperature back to homeostasis. So therefore, we are reversing the change to maintain homeostasis or stability, so that would have been B. The other answers are all about positive feedback loops, where something happens and then the response amplifies it. So it would have been like we get a change and then it will go higher and higher. These are things like blood clotting and childbirth, and the menstrual cycle hormones have a lot of positive feedback loops to them as well. Okay, let's move on to number 5. Which chemical bond forms when electrons are shared between atoms? So first off, I want to know that atoms can bond to other atoms through covalent bonds. So for example, this is CO2, where you have a carbon bound to 2 oxygens. What these lines are representing are covalent bonds, where there are basically 2 electrons inside each bond, represented by the dark circle. Those are each individual electrons. And it's as if these atoms are sharing the electrons between each other. Now, I know another question that they could ask you on a test is which atoms like to form these covalent bonds. And it's typically going to be molecules that have CHON P. All of these guys will form covalent bonds, usually between themselves. Anytime you have like minerals like sodium or chlorine or potassium, these will all form what's called ionic bonds, where they're actually transferring electrons between themselves. So be really careful about this. I know a lot of A&P teachers harp on this a lot. So be aware, covalent, we're sharing electrons between John P atoms, and then with ionic bonds, we're transferring the electrons in things like minerals like sodium, potassium, and chlorine. All right, so number 6, a solution with pH 3 is how many times more acidic than pH 6? I have to say, if your instructors ask you this, they really love their chemistry. So when we're talking about the pH scale, we know that it goes from 0 to 14, 0 being very acidic and 14 being very alkaline, or the opposite of acidic. Now, if we're talking about pH 6, whenever you move from one pH to the next, so let's say we go from 6 to 5, we know that one leap in number is adding basically a 0 to 1. So that basically means 5 is 10 times more acidic than 6. Well, when we go from 5 to 4, we know that we add another 0. So at this point, 6 to 4, if we add a 0 to 10, it is 100 times more acidic. So you get the picture. If we're going from 6 to 3, which would be the right answer? 3 times, 10, 100, or 1,000 times? It would be 1,000 times, because we're going 3 different leaps. 1, 2, 3. Therefore, we'll have 3 zeros associated with it. So from 6 to 3 would be 1,000 times. Very good. So that's a kind of a tough one. I'm not sure if they'd ask you that, but it's good to be aware of it. Number 7, which macromolecule is primarily responsible for long-term energy storage in humans? So we have the answers: carbohydrates, proteins, lipids, and nucleic acids. Now, if you're a smart A&P student, you probably learned that carbohydrates can store energy. But the key word here is long-term energy storage. So when we think about long-term energy storage, things that can stay around for a while before being broken down, we're going to say that it is lipids. So lipids are going to be long-term energy storage, whereas if you're using carbohydrates, we know that carbohydrates are going to be a little more short-term energy storage. And what will actually be used first is carbohydrates. So say you have a stored carbohydrates in your liver in the form of glycogen, and you go start running a marathon, you're gonna burn up the carbohydrates first, right? Because it's short-term energy storage. But over time, if you were to do like an ultra marathon, you might start breaking down fats for more long-term energy storage and usage, okay? Just be aware of that. Both of them store energy. This one's more short-term, that one is more long-term. So the correct answer would be C. Okay, next one is, what is the monomer of proteins? This one you kind of know it or you don't. Is it fatty acids, amino acids, nucleotides, or monosaccharides? The first one is kind of the monomer of lipids. B is amino acids. That is the monomer of proteins. I want you to know that there are 20 different amino acids. They might ask you that as well. There are 20 types of amino acids, so they're very diverse in shape and function. Okay, they're the most diverse. The third one we were saying was nucleotides. So that would be a monomer of the nucleic acids. And then monosaccharides means single sugar. That would be the monomer of carbohydrates. So just be aware of all those different ones. A&P professors like me, we could phrase these questions in a million different ways. Okay? Number 9, we're moving on. So we've got an enzyme increases its reaction rate by what? Okay, so we've got raising the activation energy, lowering the activation energy, changing the reaction equilibrium, or permanently altering substrate structure, if I can read. So enzymes, I like to think of enzymes as staplers or scissors. So imagine you have like a strong rope, okay, and you need to break that rope in half. Well, if you could really try to break that rope in half, but it wouldn't work very well unless you had scissors. So what scissors do is you could basically snip, snip, and then rope breaks apart. So what did it do to the amount of energy required to cut that rope apart? It lowered it. It made it easier for you to cut the rope apart. So the correct answer would be lowering the activation energy, because the activation energy is the energy required for the reaction to occur. So they may ask you a question about a graph where, let's say, we have some sort of inputs, and then we have an energy that's required to do it, and then we have energy in the products. This is an exergonic reaction. So energy is on the y-axis, and then time would be on the x. They would say this from here to the base of the inputs would be the activation energy. Now, whenever you have an enzyme added, a lot of the times they will show you a graph that looks more like this, where now the activation energy is cut in a very small way. So now the activation energy is only like that. Well, that would be the activation energy with an enzyme. Why? It was lowered, made easier for that reaction to occur because of the enzyme. So I know a lot of A&P professors use this graph, so just be aware of it. Okay, activation energy is lowered whenever you use an enzyme. Brilliant. Okay, moving on. Number 10, we're on to number 10 already, y'all, we're cruising. ATP is best described as a structural carbohydrate, the cell's energy currency, a membrane phospholipid, or a nucleic acid polymer. Now, the two that could get you tripped up is about the energy currency and nucleic acid polymer. ATP is actually a monomer of a nucleic acid, so it's made of an adenosine group, which is kind of a nitrogenous base, and then we get the phosphate. So it is technically a nucleotide or a part of a nucleic acid, but the best way is to know that ATP is your cell's energy currency. And we know that ATP, we've got an adenosine group here, and we've got 3 phosphates attached to it. What happens when we use ATP is we will use an ATPase enzyme, right? So ATPase, it's an enzyme that cuts ATP. We'll cleave that phosphate off. The phosphate will actually go flying, and that will be some sort of useful energy for the cell. So sometimes you'll see the following. Sometimes you'll see like a pump in the membrane of a cell. Maybe it's called like a sodium-potassium pump. And they sometimes tell you it's a sodium-potassium ATPase pump. And what you'll see in these pumps is you will have a molecule of ATP going into it, and then you see it turns into an ADP and a phosphate. Well, what the heck does that mean? I always was confused on this in college. Well, what's that showing? It's showing that that phosphate got clipped off, and now you are left with an ADP and a single phosphate by itself. That's all that's showing. So it's basically showing there's some energy investment into the system so that it can do something cool. So in this case, pumping sodium out of the cell and pumping potassium into the cell. That wouldn't have happened had we not invested the energy in ATP to do that. All right, so that's it for part 1 with the intro, atoms, and chemistry. Now we're going to go into cells, tissues, skin, and bones. And if this has been helpful so far, I need you to do a couple of things. Like this video, subscribe to the channel, and if you do those 2 things and then comment anatomy rocks. So in the comments, write anatomy rocks. You will get placed in a drawing to win some free organized biology swag. So please make sure to do that, and I'm going to ship it to 1 lucky person in the comments below, but you have to be subscribed and you have to like this video. All right, moving on. Number 11, which cell organelle is the site of oxidative phosphorylation? So we've got the Golgi apparatus, smooth endoplasmic reticulum, mitochondrion, or the lysosome. Now, you may have heard of this term before, oxidative phosphorylation. It's basically where we are using what? Oxidation, which means oxygen. We're using oxygen to do something called phosphorylate, okay? What does that mean? Using oxygen to phosphorylate something. Well, this is when we're using that molecule of oxygen in order to basically be the final electron acceptor. If you have questions about it, I've got a video called the electron transport chain. And what that will eventually do is allow us to phosphorylate an ADP molecule. What does that mean? We're going to add a phosphate to that ADP molecule. If we do that, well, what do we form? ATP. So this is actually aerobic cellular respiration, oxidative phosphorylation. Well, where this occurs is a very cool looking organelle that kind of looks like this. It is the mitochondria. So in the mitochondria, we do oxidative phosphorylation, adding a phosphate to an ADP to produce ATP. That's why it's called the powerhouse of the cell, right? All right, so that would be C. Number 12, which membrane transport process requires ATP directly? Okay, we've got simple diffusion, facilitated diffusion, osmosis, and primary active transport. Now with that, anytime you hear that word active, I always want you to remember active equals ATP. We require ATP for active transport, usually when we're doing something like pushing something from low to high, transporting large amounts of objects. The other three, diffusion and osmosis, would be called passive transport. So anytime we're talking about passive transport, that falls under the category of diffusion, which is basically things moving from high to low, and that requires no energy, okay? So it requires no energy for my marker to fall high to low, but it does require me energy to pick up the marker from low to high. That would be considered active transport, okay? Pretty straightforward and simple, so the correct answer would be D. Now osmosis, going back to osmosis, is the movement of, we got solutes from low to high concentration, water from high to low water potential across the membrane, ions through ion channels driven by ATP, and then large particles via vesicles. So osmo, anytime you hear that prefix osmo, it's usually referring to some form of water, like osmolarity is a measure of solute in water, okay? So we're talking about water moving, and osmosis is a specialized type of diffusion where we're moving water high to low. Okay, we're moving water from high to low, so therefore the correct answer, you guys can probably see, would be B, water from high to low water potential across the membrane. It wouldn't be A, C, or D because all of those would require ATP. It would be a type of active transport. So anytime high to low, it's diffusion, passive transport. Anything from low to high, we got to use ATP, it's active transport. Moving on to number 14. During receptor-mediated endocytosis, uptake is, oh, what the heck is this? Non-selective and random, selective for specific ligands bound to receptors, driven by actin polymerization only, or equivalent to exocytosis. So receptor-mediated endocytosis. Let's say we have a cell here, okay, and we're doing endocytosis. So endocytosis refers to into the cell process, basically, endocytosis, okay? So we're bringing things into the cell. Well, sometimes we have things outside of the cell that have a specific shape that we want inside. Well, in order to basically get those in, we have receptors here that will bind to that product perfectly. From there, the cell will basically get the signal, hey, this is a good thing, we want to bring it in. And the cell will bring it into a vesicle, and that vesicle will now contain that product. So if we think about it, that is a very specific product that's binding to a very specific receptor to bring it into the cell. So if you look at the possibilities, it's likely B because it's selective for specific ligands, meaning that molecule, it's a ligand, binding to receptors, okay? The other ones would not be correct because exocytosis means pushing out. Actin polymerization is proteins basically being added inside of the cell. And then it's not non-selective and random because that is a very, very specific substrate or ligand that we are binding to, okay? So that's a little tougher one. Number 15, which tissue type lines body surfaces and forms glands? Anytime you hear the word lining, no, it is epithelial tissue. That would be C. So I remember that epithelia literally translates to a sheet-like thing upon something. So epi means upon, and then thelia means sheet, okay, or sheet-like. So therefore, it is a sheet-like cellular structure upon surfaces. I always remember that if you take your finger and touch any part of your body, you are likely touching epithelial tissue because it's lining all the structures of your body. Let me erase this and we'll go to the next one. Number 16, we've got a stratified epithelial tissue is adapted for what? We've got rapid diffusion, secretion only, protection from abrasion, or contraction. So when we think about D, contraction, contraction's only gonna happen with muscular tissue. That's tissue that I can actually contract. So we're looking at 3. Well, rapid diffusion always occurs whenever we have cells that are very thin and only in one layer. So as an example, this is in your alveoli of your lungs. This is where simple diffusion will happen. So oxygen will actually go into the bloodstream, CO2 will diffuse out. That is actually simple squamous epithelial tissue where we have one layer. So this is stratified epithelial where we have multiple layers. So it can't be A. Secretion only. So this is where you got to be careful. Secretion only. As an example, we have stratified cuboidal tissue that can actually be secretory. So as an example, your sweat glands, as an example, are, I believe, stratified cuboidal, and they will start secreting sweat into the lumen of your body. Now, when the word says only, that means that that's the only function it has. However, we know if you look at the epidermis of your skin, so here's your skin, you got the epidermis, you've got the dermis and the subcutaneous. This part is made of very stratified squamous cells. So if it's made of stratified squamous cells on the epidermis of your skin, this is more for protection against abrasion. So the correct answer for this one would be C. It's adapted for protection against abrasion. It can't be B, although sometimes stratified cells do secrete. It's not the only thing that stratified cells do. It also can protect against abrasion. Okay, so that was a more difficult one. Let's go to number 17 about tissues. Which connective tissue fiber provides the greatest tensile strength? Elastic fibers, collagen, reticular, or myofilaments. Now, tensile strength basically means if you basically force on these proteins, it will slightly bend, but it'll keep its shape pretty strong, okay? So we're gonna be strong in one direction. Anytime we're talking about strength in one direction, which is tensile strength, we're going to deal with collagen fibers. So collagen, I like to think of as steel rods in a way, running through things like your dense regular connective tissue, so like your tendons, your ligaments, these really strong fibrous structures that hold things together well. So anytime you're using collagen, it's going to be very strong in one direction. You also have collagen in your bones to keep them basically upright and preventing them from kind of bending and flexing too much. Very similar to steel rods. All right. Number 18, which cell type produces bone matrix? There's a key word, produces. Anytime you're thinking of something producing something, we are always going to use the suffix blast. Blast cells always build. So chondroblasts build cartilage, okay? Hemocytoblasts build blood cells. So in this case, we're talking about building bone matrix. We're going to use those osteoblasts because those are going to be bone builder cells. Okay, osteo always means bone. Blast is a builder cell, as I just mentioned. Okay, so anytime you see blast, it's a builder. Anytime it's a clast, it breaks things down. Anytime it's just a site, it maintains. So just know those different suffixes. Number 19, hyaline cartilage is found where? We've got the intervertebral discs, the external ear, articular surfaces of long bones, or ligaments. Well, we know for A, the intervertebral discs contain fibrocartilage, which is padding between bones, like between your vertebrae, so it can't be that one. The external ear is actually made of elastic cartilage. As you can see, it's very elastic, so it's not that one. C, articular surfaces of long bones, so we know like in bone tissue, if you have a long bone like this, at the very ends of them, we will have these little protective caps on them that are made of hyaline cartilage. So the correct answer would be C. The articular cartilage contains hyaline cartilage. And then ligaments are dense regular connective tissue, so it wouldn't be D. All right, moving on to number 20. We are cruising, y'all. You're doing fantastic. The epidermis is primarily composed of, hey, y'all, check it out. We have this one. Epidermis, top layer of the skin. What's it made of? You see that it is stratified squamous, many layers, right, of thin, flat cells. Keratinized epithelium. Keratinized is basically a protein that protects against abrasion. So it would have been B. So that was an easy one. Number 21, which layer of the epidermis contains actively dividing keratinocytes? So we know the epidermis has many, many layers of flat, thin cells. But on the bottom layer here, this is where we are going to have things called stem cells. Now, stem cells are regenerative cells that will continue dividing to produce cells above them, which will flatten out, and eventually get sloughed off. But these stem cells remain. Now we notice the stem cells are on the bottom or basal layer of it. So the correct answer would be the basal cells on the bottom. So the stratum basal layer, okay? Sometimes they add that little extra E. I always think basale. It sounds like an Indian restaurant, but stratum basal layer, the bottom layer of the epidermis. Number 22, which pigment primarily determines skin color? You may know this one already. We've got hemoglobin, melanin, keratin, and bilirubin. So we know hemoglobin is actually the protein in your blood. And while oxygen amount on the hemoglobin can change your skin color, it's not going to be the primary determinant. Now, melanin, we know, is a pigment protein. It can be pheomelanin, which is going to be more of the red and white red and yellow pigment of your skin. And then we also have eumelanin, and we know that that is going to be more of the black to brown to white pigment. If you don't have any, it'll be more white. If you have more, it'll be black or brown. So that is the main protein pigment that's going to give your skin its color. However, I have to mention carotene, basically beta carotene from vitamin A in like sweet potatoes and pumpkins, and bilirubin, which is a byproduct of red blood cell recycling, also can change the color of your blood, which can then personify in your skin color. So as an example for like the first one, hemoglobin, if you have less oxygen, you'll appear a little more blue. If you have more oxygen, you'll appear a little more red, okay? Just because that hemoglobin has different amounts of oxygen. So it can affect it, blood components, but it's not the main determinant of skin color. Number 23, the primary functions of the integumentary system include all of them except for, we've got protection, vitamin D synthesis, hemopoiesis, or thermoregulation, okay? Well, I can give you a quick acronym for your integumentary system function. I always say the skin is stupid skin with 2 P's. So what do we have for skin? We have sensation, we have thermoregulation, so basically temperature regulation. We have UV protection with our melanin. We have preventing water loss, so preventing water loss like insulation. We have protection just in general. Then we've got immunity, so we're protecting from pathogens. And then we've got D vitamin synthesis. So we produce vitamin D with our skin. So therefore, which one is not involved in this question? Obviously, it is going to be, if I can find it, hemopoiesis. So that's actually blood cell synthesis. That occurs in your red bone marrow. So that occurs in red bone marrow. So the answer would be C. Number 24, long bones grow in length at the, keyword here is we're growing bone into a space, therefore lengthening the bone. We've got the periosteum, medullary cavity, epiphyseal plate, or the articular cartilage. So we know that in bones, let's say this is a bone again, if the bone has a dark, basically line inside of themselves made of hyaline cartilage, this is called the epiphyseal plate, as you can see on the question. That is also known as the growth plate because bone can only develop into hyaline cartilage. It's called endochondral ossification, into cartilage bone development. So therefore, as bone grows into the epiphyseal plate, the bone will grow lengthwise. So the correct answer would be C, the epiphyseal plate. Now, once you're done growing, that will actually turn into bone tissue. It'll calcify, and that'll turn into the epiphyseal, not plate, but it'll turn into the epiphyseal line. Okay, basically saying it's all bone now, you can't grow anymore. Okay? Next one, we've got 25. The compact bone is organized into structural units called, this is kind of a you know it or you don't. The correct answer is going to be the osteons. Okay, the osteon is a functional unit of the compact bone, whereas in spongy bone, the functional unit is going to be the trabecula. The other two chondrocytes are just cartilage maintaining cells, and the lacuna is just the space where those bone cells and chondrocytes live, essentially. So those would not be relevant. We're cruising, y'all. We're almost into the next section of joints, muscles, and the nervous system. So let's keep rolling. Number 26, which cell breaks down the bone matrix? Y'all, you should know this one already. We went through it. It would be the osteoclast, the bone breaker downer, letter C. Well done. Number 27, which hormone increases osteoclast activity and therefore raises blood calcium levels? We've got calcitonin, parathyroid hormone, insulin, and growth hormone. Once again, this is kind of, you gotta memorize it, but we know the answer is parathyroid hormone. So parathyroid hormone, okay, is released whenever we have low blood calcium levels. So if we have low blood calcium levels, what we could do is take some of the calcium in the bone and put that calcium instead into the bloodstream. So we've got low blood calcium, we release parathyroid hormone from the parathyroid glands, that goes to the osteoclasts, and it will stimulate the osteoclasts. What do the osteoclasts do? Well, osteoclasts dissolve bone tissue and therefore release the calcium in the bone into the blood. What does that do? Well, that raises your blood calcium levels. So if we're raising our blood calcium levels, we are back to normal, and it's going to be a negative feedback loop. We'll basically cut off all of this and stop all of it, and we'll be back to normal. That's called a negative feedback loop. So it would have been parathyroid hormone. Equal and opposite is calcitonin. It's released when we have too high blood calcium levels. It goes to the osteoblasts in that case and tells it to put the calcium into the bone out of the blood, therefore lowering blood calcium back to normal. The last one for this section, a fracture where the bone breaks the skin, is called what? Well, if you see the bone breaking through the skin, it's kind of open to the outside environment, is it not? So we call that guy an open or compound fracture. If you watch the Louisville basketball game where Kevin Ware basically landed on his toe and his tibia snapped, you could actually see it sticking out of his skin. Not fun, it was a compound fracture. Now let's move on to part 3, which is joints and muscles. Now, before we get into this section, I want to ask you, is this a helpful format? Do you enjoy me having the question here, writing the answers here on the board and talking about it? Or would you like it to look a little different? I'm open to feedback. This is my first final exam review, so please let me know if this has been helpful in the comments below. Or you can email organizedbiology@gmail.com and let me know as well. Number 29, which joint type is immovable? We're not moving it. We've got synarthrosis, amphiarthrosis, diarthrosis, and synovial. Well, we know the synovial joints are really movable, okay? So any movable joint is going to be synovial, so it's not that one. Diarthrosis is actually a classification of synovial joints. It's very freely movable. Amphiarthrosis is kind of partially movable, but synarthrosis gives you that key. Syn refers to together. So together arthrosis literally translates to a together joint, like the sutures in your skull or the gomphosis joints in your teeth to your mandible or maxilla. These are not movable joints. So therefore it would be A, synarthrosis. Number 30, which structure reduces friction in a synovial joint? So we've got articular cartilage, bursa, or the tendon sheath. Now, we've also got all of the above, and I would tend to say all of the above. Reason for that is the tendon sheath is going to be basically covering the tendon to prevent friction on the tendon as it kind of changes shape. The bursa is usually freely floating, like the bursa on your elbow or the bursa of your patella. And as those bones move across other different structures, the bursa is going to allow that gliding motion to reduce the friction. And then articular cartilage also is like a capping of plastic on the ends of bones. So as it glides, it'll also reduce friction. It's very similar to like a plastic cap that you put on furniture. So all of these are going to decrease the friction. And we know that as you get older, all of these three things get more inflamed, friction increases, and that could potentially lead to arthritis, which is inflammation of your joints. Number 31, skeletal muscle fibers are multinucleated because, well, first off, multinucleated basically means if you look at a skeletal muscle fiber, we've got many nuclei inside of it. So maybe like 3, 4, 5 in one muscle fiber, which is a muscle cell. Why did that happen? Let's read them. Nuclei fuse during development from myoblasts. Each myofibril has its own nucleus. Nuclei move during contraction, or they are actually mononucleated. So this question is wrong. Well, we know that there are multinucleates in skeletal muscle fibers. Do the nuclei move during contraction? Well, that wouldn't change. So as the muscle contracts, the nuclei would stay within the cell. So it's not C. Then B, each myofibril has its own nucleus. Well, you know, hopefully, that inside of a muscle cell, there will be individual tubes running through it called myofibrils. And these myofibrils basically contain all of the proteins necessary for the muscle to contract. So like actin, myosin, tropomyosin, etc. And those are just proteins. There's no nucleus in proteins themselves, okay? So the only answer that would be possible is nuclei fuse during the development of myoblasts. Well, you know, myoblasts are muscle building cells. So what happens actually is you have many cells, myoblasts, with nuclei inside of them. And as your muscle cells rebuild themselves, these guys will fuse together. And when they fuse together, they will form muscle fibers. So all the nuclei kind of come along with it when they build the muscle fiber itself. So the correct answer would be A. Number 32, the functional unit of a skeletal muscle fiber is the what? So if we look at a muscle fiber, what's the functional unit of contraction? Well, we know inside the myofibrils, there will be these repeated units that will kind of give rise to the striations. And these repeated units are literally translated to muscle units, which are sarcomeres. Okay? I always remember mere kind of means like a unit. Sarco usually refers to fleshy, but I always remember this as muscle. So it's literally the muscle unit. As those sarcomeres shorten, come closer together, we will contract the muscle. So the correct answer would be C, the sarcomere. Now the other structures are important for the contraction of the skeletal muscle, but it's not the functional unit. Number 33, during muscle contraction, which filament slides past the other? We've got thick filaments slide past the thin filaments slide past the thick, both shorten equally, or neither, only the sarcoplasm changes. This is a classic question just trying to throw you off. We know that muscles contract with the sliding filament theory where we have a myosin thick filament. So myosin is very thick and you can kind of see that it is. And then you've got the thin actin filaments surrounding it. So these are the thin actin filaments or myofilaments, okay, so thin. And when we contract the muscle, the myosin heads will grab the actin and pull it inwards. So therefore the actin will slide that way and slide that way and slide that way and slide that way, according all these Z lines closer together. So therefore, does the myosin ever move? Not really. The thin filaments are gliding past the thick. So the correct answer for this one, if I can find it again, is the thin filaments slide past the thick filaments. It would be B. Very good. Number 34, calcium ions released from the sarcoplasmic reticulum bind to what? We know that calcium is the key to contraction, and it binds to troponin, and it helps pull the tropomyosin head off to allow myosin to bind to actin. If you want to learn more about that, you can watch a sliding filament theory of contraction in my YouTube channel. So the correct answer would be B. That's what it would bind to directly. 35, ATP is required during muscle contraction for the following. A, cross-bridge formation only. B, cross-bridge detachment and power stroke cycling. C, calcium release only. Or D, maintaining membrane potential only. Well, when we're talking about ATP is actually going to bind to the myosin head. So what happens is here's the actin and here's the myosin head, and it's cocked back. At this point, ADP and a phosphate are actually already attached to the myosin head. That implies that we had some energy invested in that myosin. Now, when that calcium comes in, binds to troponin, pulls tropomyosin off, we can form what's called a cross bridge. So now myosin grabs the actin. At that moment in time, it will actually drop off the ADP and a phosphate, and the myosin will pull that way, thus pulling the actin centralized, right? So like pulling it towards the M line, the center. Now, once that occurs, the myosin head is like a catapult. We just threw it, and now it's all weak. So we need to add another ATP to it. So here's the myosin head weak. We need to add an ATP to it so that we can re-energize it back into its energized state. So now ATP, ADP, and a phosphate was cleaved here. Why? Because we use that energy to pull the myosin head back. Well, when that happens, the myosin head detaches from the cross bridge, which is basically when the myosin binds to the actin. So it does a couple of things here. It has a cross bridge detachment when ATP binds, the myosin head pops off of the actin when ATP binds to it, and power stroke cycling. Power stroke cycling basically says once myosin grabs and pulls, it's got to bind back from ATP, bind back to actin, and then pull again. That's called power stroke cycling. So ATP is both used in the detachment as well as in the cycling of it. So that would have been answer B. That was a tough one. Number 36, the twitch summation in tetanus refer to what? Types of muscle fibers, patterns of muscle contraction force generation, steps of the neuromuscular transmission, or the bone remodeling phases. So what this is showing is basically when the muscle fiber is stimulated by a neuron. So we've got the twitch, where the muscle fiber is contracted for a quick second and then it goes down. We've got summation, where we're starting to recruit more and more motor units. So what you might see on a graph is the muscle's voltage. So this is the muscle's voltage. Basically, when it gets positive, we're contracting. If we have a muscle twitch, it will look like this. It'll twitch, be contracted, and then stop. If we're doing summation, we're starting to recruit more and more muscle fibers. So it might look something like this. Or if we're in tetanus, we're fully contracted and we're staying positive for a long period of time. So this would be a twitch, this would be summation, then this would be considered tetanus. So therefore, what are we looking at here? We're looking at patterns of muscle contraction force generation in 3 different scenarios. So this is fully contracted, pretty strong. This is summation. We're starting to contract, starting to have more force. And then a twitch is we've got a force, but it's very short-lived, and then it gets weak again. Okay, so it would have been answer letter B. Okay, few more, and then we're into the part 4 nervous system. 37, which muscle fiber type is most fatigue resistant and high in mitochondria? Remember previously we talked about mitochondria being oxidative phosphorylation. We require oxygen for it. So we know it's going to deal with oxidative something, right? So the only thing we have here is B, fast oxidative, or C, slow oxidative. Now when we look at most fatigue resistance, if you are fatigue resistant, okay, are you doing something really, really fast? Or are you doing something really, really slow? In this case, it's going to be C, slow oxidative, because slow oxidative muscle fibers are very long lasting. Like your back, like your legs, you can contract them for long periods of time without becoming fatigued because we're doing a lot of aerobic respiration, slow oxidative aerobic respiration. Very good. Number 38. Motor units with few muscle fibers per neuron allow for, okay, so we've got A, strong, powerful contractions only, B, fine control and precision of movement, C, fatigue resistance exclusively, and D, automatic reflexes only. So you may know the answer to this. If you don't, let me draw what a motor unit is. I know I taught this in my classes. A motor unit is basically talking about you've got a neuron, okay, a motor neuron that's going to come in and communicate to a specified number of muscle fibers. So let's say an example, this neuron is going to come and contract like 3 muscle fibers, okay? So here's muscle fibers, and this neuron, when stimulated, sends an action potential, stimulates these 3 muscle fibers to contract. This would be considered a very small motor unit because one neuron controls a very small number of muscle fibers. We have other motor units where one neuron can have just so many different branches and basically innervate many, many hundreds to thousands of different muscles, okay? Of muscle fibers, I should say. So here's a bunch of muscle fibers being connected by one neuron. We would consider this a large motor unit. Now, if you think of, in this case, what did it ask for? It asked for motor units with few muscle fibers. What does it allow for? It would be B, because if we have just one neuron controlling literally microscopic cells, just a few of them, we can do very fine motor, precise movements. This is going to be like in your hands and your face. Whereas the large motor units are going to control like one neuron to like thousands of muscle fibers, like a big old crane. You've only got a few wires, but it's moving these big, gross movements. These would be things like your back and your legs. Big motor units, just one neuron controlling many of them. So the correct answer would be B. Number 39. The neuromuscular junction uses which neurotransmitter at the motor end plate? This is pretty much you know it or you don't. When we're looking at this neuron communicating to the skeletal muscles, we have a specific neurotransmitter being released, the chemical from the neuron, to talk to the skeletal muscles. We know this is called acetylcholine, and it's going to stimulate these muscle fibers eventually to contract. So the correct answer would be C. Number 40. Which structure carries action potentials into the interior of the muscle fiber? So first, action potentials are basically saying, hey, when does this muscle get positively charged? And then how do we share that throughout the muscle fiber so that the whole muscle fiber contracts at the same time? Well, we know throughout the muscle fiber there are these little tubes that are going to be carrying all of those positive charges all throughout that muscle fiber itself. That is called a T-tubule. It'll tell all of the muscle cell to get positive immediately at the same time to fully contract. So therefore, the correct answer would be A. All right, let's move into the nervous system now. All right, so I know this is the part of the video. It's been a little while. You're in number 41 now. So I'd just love for you guys to just take a big deep breath and breathe out. This is what I want you to do during a test. It can be stressful. Your brain can start going to mush. Just take a breath, look around for a second, breathe deeply, and your brain will reset. So do that during the test. It'll help you tremendously. Now let's keep rolling. Number 41. The central nervous system consists of the, what is it? Brain or spinal cord, cranial nerves only, spinal nerves only, or peripheral ganglia. This one's a pretty simple one. Central nervous system, the main structures are your brain and your spinal cord running down the center of your body. So the correct answer would be A. Number 42, which glial cell or supporter cell produces myelin in the central nervous system? We know in the central nervous system there are several different glial cells. Now they all don't start with the letter S. So I want you to remember that the CNS glial cells have no S word glial cells. So therefore the Schwann cells are out. So we're left with oligodendrocytes, astrocytes, and microglia. Oligodendrocytes is the correct answer, so that's B. That's because they are going to wrap their membrane around those axons and produce that myelin. The astrocytes would actually serve as basically the blood-brain barrier of neurons, so preventing bad things from the blood getting into the neurons. They also support the neurons. And then lastly, the microglia are going to act as immune cells of the central nervous system, fighting off infection and the inflammatory response anytime you get damage to the central nervous system. So the correct answer would be B. Number 43, the resting membrane potential of a typical neuron is closest to which of the following? We got positive 40, 0, negative 70, and negative 200. Anytime we're looking at the voltage of a neuron, the voltages you need to remember that you will be tested over, I guarantee it, is the resting, threshold, and then action potential. The resting potential is here, and it's very negative, but it's only at about negative 70 millivolts. The threshold potential is about negative 55. If we reach that point, we send an action potential, which is the positive 10 to positive 30 millivolts. And again, that's called the action potential. This guy is called the resting membrane, or sorry, this one's called the threshold potential. My bad. Okay, so those are the ones that you need to remember for your tests. Very good. I'm flipping my page. We're on to number 44. Okay. During an action potential, the membrane depolarizes due to what? Okay, so action potential, what are we doing? We're depolarizing. That means getting very, very positive. What is actually happening in the neuron at that point? Well, we know the neuron will depolarize anytime it gains a specific ion that's very high outside of the cell, which is sodium. So when channels open up for sodium to flow high to low, sodium comes in and the neuron gets positive. Now, equal and opposite when we make the neuron negative, so when we repolarize downward, so when we're going down, we're getting more negative, that's because we're actually going to be taking a different ion, which is going to be potassium, inside the cell. We're actually going to send it out of the cell, and that makes it negative. So anytime we're going up, we're basically saying sodium in. Anytime we're going down, we're saying potassium out to make it negative. You will definitely get some questions on that. I know I will put some in my final exam for my students. So students, if you're watching, be prepared for those ion questions. All right, so the correct answer would have been what? It would have been C, influx coming in of sodium. Number 45, the all or none principle states that what? A, action potentials vary in amplitude with stimulus strength. B, a threshold stimulus triggers a full-sized action potential. C, subthreshold stimuli produce larger responses than threshold stimuli. Or D, action potentials can be partial. What did we see here? When we reached threshold potential right here, what happened? We sent the action potential. That's what's considered the all or none. So the correct answer would be B, a threshold stimulus. We get to the threshold, triggers a whole action potential, getting super positive. Very good. Number 46, saltatory conduction occurs in axons that are A, unmyelinated and short, B, myelinated and long with nodes of Ranvier, C, only in peripheral nerves, or D, found exclusively in the autonomic nervous system. Saltatory conduction occurs when you got a really long neuron's axon and you've got these wrappings of myelin sheaths around it, and therefore the action potential, when it gets sent, basically hops to things called the nodes of Ranvier. These are the nodes of, let's just put R here and here. And so therefore, if you look at the answers, we would say it would be B, myelinated and long, and they have nodes of Ranvier. So that would be saltatory conduction, where we're basically hopping those action potentials all the way to the end of the neuron. Brilliant. We're almost halfway done with a 100-question multiple choice test. You guys are crushing it. To move on, number 47, the primary sensory cortex is located in the frontal lobe, parietal lobe, otherwise known as the postcentral gyrus, temporal lobe, or occipital lobe. Well, we know that the frontal lobe deals more with personality, movement, speech. B, parietal lobe always deals with general sensation. So the correct answer would be B. But then the temporal lobe deals with like hearing and smell, as well as some memory. And then D, the occipital lobe, deals with vision. So depending on the question, you might be asked a different section of the cerebral cortex structures. Number 48, we're cruising. Which brain region coordinates voluntary movement and balance? The key word here is voluntary movement. Anytime we're talking about voluntary movement, we're going to be talking about the upper region of the brain, specifically in the outer cortex, and that's going to be called the cerebrum. Now that being said, we know we've got also like the diencephalon, which we'll talk about in a second, and then we've got this little mushroom-shaped part in the back. This is called the cerebellum. Now the cerebellum can coordinate basically unconscious movements and like procedural memory, things like when you're brushing your teeth and you're not even thinking about it. The cerebellum is actually involved very heavily in that, as well as in balance. So the key word here is it's a voluntary muscle movement, like, oh, I want to lift my bicep. Boom, that's coming from the cerebrum. Now, if it's like balance and coordination, that's going to be a little more cerebellum, all right? Moving on. The blood-brain barrier is formed mainly by what? A, fenestrated capillaries. B, tight junctions between endothelial cells and astrocyte end feet. C, myelin sheaths. And D, microglia. Well, we know microglia are like the immune cells of the central nervous system, so they're not that. Myelin sheaths are formed around the axons. There's no talk about the blood-brain barrier. A, fenestrated capillaries are basically capillaries, blood vessels, that have big open gaps inside of them. Okay? So in that case, if the capillaries are fenestrated, they have these openings, well, in that case, the blood would like just seep out onto our neurons. So the correct answer is B, the astrocyte end feet, the tight junctions between endothelial cells. So basically, we've got the capillaries with endothelial cells. These are the endothelial cells here. So they'd be very thin and flat. What happens is your astrocytes will form tight junctions between all of those endothelial cells, so the blood, and the neurons. We call this structure here that we're forming the blood-brain barrier. The reason for that is so that we can prevent toxins and metals and other things that could be in the blood from getting into the neurons, which are very sensitive. We don't want to damage those neurons, so the astrocytes basically select what can come out and what stays away. Very good. Okay, number 50. Wow, we're on halfway there. SpongeBob reference. I'm already halfway there, halfway there, halfway there. I love SpongeBob. Anyway, moving on. Which cranial nerve is purely sensory for vision? So point out here, cranial nerves can contain either sensory information alone, motor information alone, or sometimes both, depending on the cranial nerve. So in this case, we're just talking about sensory for vision. We've got the optic, oculomotor, trigeminal, or facial. If you look at the words, optic always refers to sight or vision. So optic, cranial nerve number 2 would be the correct answer, A. Moving on to number 51. Autonomic nervous system efferent pathways typically have what? I love this question. A, one neuron from the central nervous system to the effector. B, two neurons, preganglionic and postganglionic in series. C, three neurons in a chain. Or D, no synapses outside the central nervous system. Well, we know the autonomic nervous system, it's efferent. So basically what we have is in the central nervous system, we're going to go away from it. We're going to affect some tissues. In either parasympathetic or sympathetic, what you'll see is you have a neuron here. It'll go to something called a ganglia, talk to another guy, and then he will actually communicate to, let's say, like the heart tissue to change its heart rate. Well, we know we've got a two neuron system here, a pre-ganglionic and a post-ganglionic, before the ganglia, after the ganglia. So therefore, the correct answer would be B, two neurons, pre and post-ganglionic in series. All right, here to help me with number 52 is the sympathetic... Sorry, this is Gracie. I didn't introduce her properly. Number 52. The sympathetic nervous system generally does what? A, promotes rest and digest responses. B, promotes fight or flight responses. C, decreases heart rate and dilates pupils. D is the same as the parasympathetic system. Okay, so demonstrating is this dog of mine. This is Gracie. And I picked her up and she started getting stressed. And so I felt her heart rate increase and I felt her get stressed out. So we can remember that the sympathetic nervous system promotes fight or flight responses. Right now she wants to flight because she's not about to fight me. She's too cute and she likes me too much. All right, we're gonna move on from there. All right, so now we're kind of getting into the sensory division of the peripheral nervous system. So sensory receptors that detect stretch in muscles are called the Pacinian corpuscles, muscle spindles, Golgi tendon organs, or Ruffini endings. So A and D are both located in the skin, so basically feeling things on your skin. So it's got to be B or C. And with stretch in muscles themselves are going to be the spindles wrapping around different muscle fibers so that therefore we can detect their stretching. The Golgi tendon organs are located in tendons, and they're going to detect basically pressure and kind of pulling of the tendon. So in muscles, it's going to be B, muscle spindles. Number 54, which type of receptor is rapidly adapting and detects vibration and pressure? So with this one, you have to read the answers carefully. We get the Merkel discs, which are slow adapting, Meissner's corpuscles, rapid adapting, free nerve endings, and chemoreceptors. So chemoreceptors detect chemicals, which your skin can't really feel. We're kind of talking about vibration and pressure. The free nerve endings of your skin are going to be kind of near the epidermis. So here's the epidermis of your skin. And these are going to be the free nerve endings kind of coming up. And these a lot of the times detect pain. This is how I normally write, right? With my hand like that. So with pain, you don't really want it to be adaptive. You actually want to continuously know about pain. So we're not going to adapt to painful stimuli very well. So therefore, it's the Meissner's corpuscles or the Merkel discs. With Meissner's corpuscles, anytime you see those guys, Meissner's, this is going to detect 2-point discrimination. Basically knowing, is one thing touching you or is multiple things touching you? So these guys, as it's mentioned, are very rapid adapting. So which one is rapidly adapting and detects vibration and pressure? With these guys, this is just pressure. We're feeling 2 different points. Like say you have a little prick there and a prick there. The Meissner corpuscles down here will be able to detect multiple points of pressure on the skin, but they do not detect vibration. So the correct answer will be those Merkel discs. They're going to be kind of a little more superficial and they can detect those vibrations and pressure. So that would be A. Number 56, the chemical synapse releases neurotransmitter into the what? Well, if we're looking at those answers, we get the synaptic cleft, we've got the axoplasm, myelin sheath, or perineurium. So there's a lot of words there that can kind of throw you off. You're like, that kind of sounds right, correct? But know that anytime there's a neuron communicating to something, we have the axon terminating here, and then there will be a space in between the axon and some sort of target cell. This space is called the synapse, and so therefore the best answer would probably be something dealing with the synapse, which would be the synaptic cleft, which is basically the invagination inside of that synapse. So that would be the correct answer in this case, A. Okay, number 56. Which ion entering the axon terminal triggers neurotransmitter release? Well, we can use this picture, and we know inside of the neuron we've got these vesicles containing the neurotransmitter, and we want to release them into the synaptic cleft, or the synapse in this case. So what different channels will we open? Well, we know we will open up voltage-gated calcium channels, because calcium likes to move stuff when it comes into the neuron. It's going to push out those vesicles, releasing the neurotransmitter. So anytime we're talking about moving something, it's going to be dealing with calcium. So calcium pushes out vesicles. It moves the tropomyosin off the myosin binding sites in your muscle cells. So calcium always likes to move things. Number 57. So that would have been, C. 56 would be C. Number 57, in reflex arcs, in interneuron, A is always absent, B connects sensory motor neurons within the CNS, C carries signals to effectors outside the CNS, or D is a type of effector organ. Well, the interneuron we know is found inside the spinal cord. So whenever we're doing a reflex, what we know is there's a sensory neuron sending information into the central nervous system. And then what happens is we're going to bypass the brain altogether. So we're going to have a motor neuron here that's going to go out and communicate to some sort of effector organ. Maybe it's a muscle. But in order to do that, we have to basically coordinate these to make the sort of arc. That arc is completed with what we call an interneuron. So that would be this guy here. And he's between two neurons, one here and one here. That's why he is called an interneuron. So the correct answer would definitely be the B, connecting sensory and motor neurons within the central nervous system itself. Number 58, which brainstem center regulates basic life functions like heart rate and respiration? This is kind of a you know it or you don't. We've got A, thalamus, P, the pons and medulla oblongata, C, the basal nuclei, and D, the cerebellum. Well, they're trying to trick you here because we know the basic life functions is in the brainstem, right? That's in the question. But the brainstem consists of three major parts. We've got the medulla in the most inferior, medulla oblongata. We got the pons, then we got the midbrain. So therefore, the correct answer would be B, pons and medulla, because both of these guys contain neurons that are going to deal with those basic life functions like respiratory rate as well as heart rate. 59, which nervous system structure is the major control center for endocrine functions in autonomic regulation? We've got the thalamus, A, B, hypothalamus, C, pineal gland, and then D, the pituitary stalk. So be careful here. It's talking about the nervous system structure, and we know inside of the brain, okay, this is part of the nervous system, right? There's a region of the brain down here, about right here, if this is the anterior and this is the posterior side. This is going to be called the hypothalamus. I always remember the hypothalamus deals with H, homeostasis. Underneath the hypothalamus, though, is the pituitary gland. And we know that the pituitary gland is the master regulator gland of all or most all of your hormones. So that is right here. The pituitary stalk is the connection between the two. So this is the stalk of the pituitary gland. And although it contains the axons of certain neurons coming from the hypothalamus is technically the part of the nervous system that will influence the pituitary gland. So the correct answer in this case would be B, always. All right, so now we're into the last part. The last several bit of questions is going to be kind of a catch-all. So this is going to be rapid fire, random questions that you could find on your test. Again, at this point, if you haven't already, download this sheet so you can practice the test. If you need tutoring, if you would like help from me directly, please sign up for that. And if you need any other resources, check out that resource list in the description and comments below. Now let's finish this up. This is the part of the test that gets hard. So again, take a deep breath and let's keep rolling through it. Number 60. The extracellular matrix of nervous tissue is unique because it, A, contains abundant collagen like connective tissue, B, is minimal compared to the other tissues and has specialized components, C, is identical to that of bone, or D, is composed mainly of keratin. Now, when you look at nervous tissue, you primarily will just see basically neurons, and then you'll also see these supporting cells like glial cells, like the oligodendrocytes and the astrocytes and the microglia. And so therefore, the extracellular fluid will be all the space around it. If you notice, there's really not a lot of other tissue there. It's primarily just the glial cells and the neurons. So we can know that it's not going to be composed mainly of keratin. That's going to be more in the skin. C is identical to that of bone. Bone is very hard. That doesn't make sense. B is minimal compared to other tissues and has specialized components. That would probably be the best answer because, again, there's specialized glial cells surrounding the neurons, and there's not a whole lot else, maybe some cerebrospinal fluid in there. And it wouldn't be A because it doesn't have a ton of collagen like connective tissue, because otherwise it would be super strong in one direction, which it really isn't. It actually has to be supported by other structures. So the correct answer would be B. Number 61. Which type of epithelial tissue would you expect in the small intestine for absorption? We've got simple squamous, simple columnar with microvilli, stratified squamous, keratinized, or transitional epithelium. Well, when we're talking about absorption, the biggest thing we can do to help us absorb things is increase the surface area on the cells themselves. So what we'll see in the small intestine is, like, say this is the tube of the small intestine, okay, food's coming through here. The cells that will be lining it will likely look like this. They'll be these tall cells with these projections here called, as you can see, microvilli, literally translating to tiny finger-like projections on the cells themselves. And as you can notice, this is actually a columnar cell. I didn't draw it very well. And these guys will have a variety of proteins on their tips so that they can bring those nutrients through them and into the bloodstream that would be right below it. So the correct answer in this case would be B. Now you might think it would be A, simple squamous for like diffusion of the nutrients, but in this case we don't want just anything passing through. We want only the things that these cells determine right, and they also will have the microvilli to increase the surface area for absorption. So let me erase this and we'll get to number 62. Okay, so this is kind of an easy one. It says, which staining or structure pair best identifies elastic cartilage? So we're talking about cartilage that has some elastic fibers in it. So we got A, abundant collagen in the cuna. That doesn't sound right. B, elastic fibers visible in the matrix and a flexible shape. That's going to be the correct answer, B. C, no chondrocytes present. We know that cartilage has chondrocytes, so that wouldn't be true. And then lamella arranged in osteons, that's in the bone. So that's an easy one, B. Number 63, bone remodeling responds to mechanical stress primarily by A, changing osteocyte numbers only, B, increasing osteoblast activity in stressed areas, C, only endocrine signals, not mechanical signals, and D, producing more cartilage. So we need to know that this is in bone, so we can't produce more cartilage in bone. It wouldn't be D. C, it says only endocrine signals, so maybe there's some hormones produced, but maybe not mechanical signals. Well, in this case, we're getting mechanical stress on the bone, so there's going to be some signaling there. So therefore, it's between osteocytes and osteoblasts. When we're building bone in response to mechanical stress, keyword, we're building bone, we're going to be using those osteoblasts, and they will actually build the bone matrix in concordance with the stress that is being pushed onto that bone. It's actually one of the reasons exercise makes your bones stronger in the direction, in the force at which you're doing the exercise. So the correct answer there would be obviously B. Very good. 64. Which vitamin is essential for collagen synthesis and deficiency causes scurvy? Keyword, or key letter I should say, C in collagen. So vitamin C is a very important component in order to produce collagen, and it prevents scurvy. If you ever remember the Flying Dutchman in SpongeBob, another SpongeBob reference, he's eating a bunch of fruit and he's like, fruit prevents scurvy. And we know that a lot of fruit has vitamin C in it to help us to produce the collagen. It's also important in all of your immune responses. So the correct answer would be B, vitamin C. Vitamin A usually deals with eyesight. Vitamin D is actually a hormone that does a variety of different things, helps you build bone. And then vitamin K is actually a very helpful clotting vitamin. It helps your blood clot. So if you have a question about any of those vitamins, that's what you would see. Number 65, red marrow is the site of, we talked about this earlier, A, fat storage only, B, hematopoiesis, C, calcium storage only, or D, articular cartilage production. Red marrow, again, is in the center of flat bones and irregular bones. So if you were to look at maybe like your hip bone, I can't really draw a hip bone, there's your hip bone. If you were to cut inside of it and look inside, we would see a lot of spongy bone. And in between it, we would see a lot of red bone marrow. Now, the red bone marrow contains a bunch of blood stem cells. They're called hemocytoblasts. And these blood stem cells are going to produce basically all of the blood cells of your body. So the correct answer would be B, blood cell formation, otherwise known as hematopoiesis, blood cell creation. Awesome. Number 68, fast glycolytic skeletal muscle fibers primarily generate ATP through, well, we have to look at that word, fast glycolytic. Glycolytic is basically referring to the process of glycolysis, where we are taking a glucose molecule, which is composed of 6 carbons, and what we're doing is we're actually splitting it down the middle to create pyruvate. Well, in that process of doing it, there's several chemical reactions. We actually produce 2 ATP in the process. So we're going to be looking for glycolysis, breaking apart to produce ATP. So let's see what we got. We've got A, aerobic respiration. Not that, I didn't mention oxygen here. Aerobic is with oxygen. B, anaerobic glycolysis. Did we mention that? Absolutely. Glycolysis is probably the right answer without oxygen making ATP doing glycolysis. C, oxidative phosphorylation in the mitochondria. That deals with oxygen again, making a lot of ATP. And the creatine phosphate exclusively, that is actually a way we can produce ATP, but it's going to be using creatine and creatine phosphokinase, a different enzyme. So that's a different pathway altogether. So the correct answer would be B. Number 67, which receptor on skeletal muscle binds acetylcholine at the neuromuscular junction? So we know that those neurons come in and they're going to communicate to muscle cells. Now those muscle cells will have these little receptors on there, okay? And when those muscle cells receive the signal acetylcholine, they are going to open up these little ion channels called ligand-gated sodium channels. So here's the acetylcholine bound, and then we're going to have sodium flowing in, okay? So think about that when we're reading these answers. We've got A, the muscarinic ACH receptor, B, the nicotinic ACH receptor, ionotropic, C, the beta adrenergic receptor, or D, the NMDA receptor. Now, you may not have even heard of some of these receptors before, but if you look at B, we see that it is an ionotropic, right? Ionotropic receptor. Anytime you're talking about an ionotropic receptor, look at the word ion. It's allowing ions to flow in. What's an ion? A charged atom like sodium. So even though you don't know some of these receptor types, you can see ion. Oh, that's a charged sodium atom, right? That's one example of an ion, and it's using that sodium to get the muscle positive to contract. So the correct answer would be B. Okay, the muscarinic ACH receptor is actually in your parasympathetic neuron target tissues. And the C, the beta adrenergic receptor, it's one of the ones in actually the sympathetic target tissue. So different than the somatic motor. And then the NMDA receptor, to be honest, I never talk about. So we're not even going to discuss that one. Moving on to 68. The sliding filament model predicts that during contraction, ooh, this is a good one. A bands shorten and I bands lengthen. B, the Z discs move closer together and the sarcomeres shorten. C, the thick filaments shorten. Or D, the sarcoplasmic reticulum lengthens, okay? So first off, A bands shorten. Let me draw this real quick. So we know that we have a sarcomere here, and we know that there's myosin, thick filament in the center, and then we've got the thin actin filaments on the side. Now, if you were to actually look inside of the muscle itself, what you would see is there's a lot of proteins on this section. So this section of the muscle would look very, very dark. We call that the A band, and you can say here's another A band, right? Now, when the sarcomeres shorten, those A bands, those dark sections, do come closer together. So therefore, if you look at, number 68, A bands shorten and the I bands lengthen. Now here's the key. The I bands are these guys, the light bands. So basically from here to here, this is the I band, and from here to here is the A band. So yes, the A bands kind of came closer together, but the I bands did not lengthen, they actually shortened a little bit. Okay, so when you see the next one, there will also be an I band here. So the I band will also be very light, and they're actually coming closer together. So it wouldn't be A. Then we look at B. The Z discs move closer together and the sarcomeres shorten. Well, is that true? Well, we had the Z discs here, and when we contracted the muscle, they brought closer together, right? So the sarcomeres shortened. So that would actually be B. So that's the correct answer. But then C, the thick filament shorten. The thick filaments can't shorten by themselves. And D, sarcoplasmic reticulum lengthens. Sarcoplasmic reticulum just stores calcium, has nothing to do with in and of itself contracting. So the correct answer would be B. Awesome. 69, which ion is stored in the sarcoplasmic reticulum and released to trigger contraction? I said released to trigger contraction. What's the key to contraction? Calcium. So the correct answer would be C. Sarcoplasmic reticulum releases calcium so that the muscle can contract. Number 70. Muscle fatigue during prolonged activity is also associated with what? A, increased ATP availability. B, the accumulation of metabolic byproducts and a reduced pH. C, the immediate depletion of actin and myosin proteins. Or D, the increased motor unit recruitment only. These are good. Okay, so A, increased ATP availability. If we have more energy, chances are we can do more work. So that's not going to make sense with fatigue. B, the accumulation of metabolic byproducts and a reduced pH. We do know that we produce lactic acid or lactate, reduces the pH slightly, and maybe we're making some byproducts like CO2, so maybe that's a good answer. C is the immediate depletion of actin and myosin. Well, we know actin and myosin are the proteins, but we know that they can keep cranking out and keep cranking out and keep cranking out. So it's not that they're going to be depleted, the proteins themselves, probably not C. And then we've got increased motor unit recruitment. Well, if we're increasing how many motor units we're using, that means we can use more muscle fibers, so therefore we can do more work. So the correct answer would probably be B, the accumulation of metabolic byproducts and a reduced pH. Awesome. Okay, so if you've been following for a little bit, you've noticed that all of the answers in about the last 15 were all B. Now, I want to mention this because a lot of instructors, me included, sometimes have a string of questions where I have the same letter for the multiple choice as the answer. Now, why do we do this? It is to try to force you to think, is this really the right answer or not? And a lot of the times, they are the right answer. There might be a string of 8 or 9 of one letter in a row. So just be wary of that and be confident in your knowledge. Don't worry about what letter has shown up the most, okay? So let's move on to number 71. The peripheral nervous system consists or includes what? A, the brain and the spinal cord, B, the diencephalon, C, the cerebral cortex only, or D, the cranial nerves in the spinal nerves. Remember, the peripheral nervous system is outside of the central nervous system. So we know the brain and spinal cord, the cerebral cortex, and the diencephalon are all within the brain and spinal cord. Therefore, the correct answer would be, obviously, D, the cranial and spinal nerves containing sensory and motor neurons. Number 72, which structure contains ascending and descending tracts connecting the brain and the body? I don't really like that question, but we got A, cerebellum, B, brainstem, particularly spinal cord and medulla, C, basal nuclei, or D, the corpus callosum. So the corpus callosum actually connects the hemispheres of the brain. The basal nuclei moderates motor coordination in the brain. So therefore, we're left with the cerebellum and the brainstem. We know the cerebellum deals with voluntary kind of movements and posture and balance. So therefore, the only correct answer would be B, the brainstem. And it contains these tracts going up and down, coordinating between the brain and the spinal cord. So the correct answer would be B. Number 73, a dermatome. Dermatome is, when we know it refers to the skin, because derm, we've got A, a region of the skin innervated by a single spinal nerve, B, an embryonic skin layer, C, a synonym for the epidermis, or D, a type of mechanoreceptor. Well, the dermatome, if you have like your torso as an example, you may have seen this mapping where you have like your body. Oh man, I look like a puzzle piece. There we go. So here's your body, very oblong because I can't draw on the spot. Well, you'll have these different maps like this. And these are all called dermatomes, where it's basically saying, hey, this region of your skin and region of your body is innervated by a single spinal nerve. So example, you might see like, I don't know, T1 or like C7 or C6 throughout them. And that is just referring to the spinal nerve that is associated with that region. Okay, so the correct answer in that case would be what? A region of the skin innervated by a single spinal nerve. This is also the reasoning behind referred pain. So like if you have, for example, chest pain, it feels like your chest is hurting, but it's actually your heart that is being damaged. Let's move on to number 74. The dorsal root ganglion contains A, the cell bodies of motor neurons, B, autonomic ganglion neurons, C, glial cells only, or D, cell bodies of sensory neurons. Well, we know whenever we enter the spinal cord, so here's your spinal cord, we've got two roots. We've got one side on the dorsal side, and then we've got one on the ventral side, and they're going to converge and form your spinal nerve. So it kind of looks like this. Again, I can't draw on the spot. So there we've got our spinal nerve, and then we've got the ventral front side, and then the dorsal posterior side. And we know that all of the neurons coming in on the dorsal side are going to be our sensory neurons. So in the dorsal root ganglion, right here, root ganglion, basically is a collection of cell bodies of sensory neurons. Now, if you asked what's the ventral side, what's the ventral root contain? Well, this contains your motor neurons that are going out. So that would be motor if we're talking about the ventral side, the ventral root of the spinal cord. Brilliant. Let's move on to number 75. Which reflex helps maintain upright posture by producing a rapid extension of limb muscles, especially your back? We've got the A, withdrawal reflex, B, the crossed extensor reflex, C, the stretch or myotatic reflex, or D, the Golgi tendon reflex. Well, the withdrawal reflex is basically when we're touching something damaging and we quickly pull it away. That includes flexion sometimes, so we're talking about extension here, so that's not right. We've got B, the crossed extensor reflex. Basically, if I step on something with one leg, I basically have to contract my quad on this, contract my hip flexors with this one, while simultaneously contracting my quad on the other one. So that's a crossed extensor. We're working two sides. So that's not right because, again, it's not keeping our posture. Then we've got C, the stretch myotatic reflex. This actually means basically the muscle holding steady reflex, and that's called the stretch reflex, and this is the one that's going to help us with posture. Whereas the Golgi tendon reflex, D, is basically when your doctor taps your patellar tendon and your quad kicks out in response. So the correct answer would be C for 75. Number 76, which lobe of the brain is primarily responsible for visual processing? I always remember for this one, visual is also known as optical, right? And if you write that word optical out, it's kind of a word scramble, and you can actually make close to the word occipital. So we remember that the occipital lobe is going to be the main site for visual processing for number 76, so that would be B. Number 77, which structure relays the most sensory information to the cerebral cortex? I always mention this as the relay center in your brain. We've got A, thalamus, B, hypothalamus, C, midbrain tectum, or D, the medulla oblongata. Well, we know the thalamus is that relay of sensory information, so I always draw where we have the brain and the spinal cord. Any ascending tracks coming up is usually going to go to the thalamus first, before getting basically sent upwards to the cerebral cortex to a variety of different regions. The hypothalamus, as we talked earlier, is main regulator of homeostasis. Midbrain tectum has some reflexes kind of coordinating with some cranial nerves. And then the medulla oblongata is basically the region that deals with heart rate, respiratory rate, that type of thing. So the correct answer would be A. Number 78, we're cruising. Long-term potentiation is a cellular mechanism underlying, I have a video on this if you want to watch it, dealing with memory formation. So the correct answer is B, memory and synaptic strengthening. Long-term potentiation is basically when you use the same pathway over and over and over and over again, and that neural connection becomes really, really strong. It's just like, as an example, if you have a certain memory, maybe like your wedding day that you remember really well, you've thought about that so much and had so many emotional responses to it that it's such a strong memory, you will remember it forever. You also have long-term potentiation as an example for procedural memory. So say like brushing your teeth, putting your shoes on. You've done that so many times that it's almost automatic because these circuits, these neural circuits that are being used are very, very strong and fast. So the correct answer would be B. Let's move on to number 79. The limbic system is most involved in A, the visual acuity, B, blood pressure regulation only, C, motor coordination only, or D, emotional responses in memory formation. Well, the limbic system, I always remember it's got that kind of M to it. Limbic is dealing with emotions, okay? Emotions as well as memory, okay? This is activated anytime you have some sort of heightened emotional experience. So let's say, as an example, you ate some really bad food and you had this emotional reaction because you started vomiting it up right afterwards. Well, a lot of the times you have that core memory basically burned into your memory banks, so you don't like that food for a long time. Or take a positive spin on it. Your wedding day, right? Heightened emotion, you're really happy, and so that also burns in your memory. So the limbic system basically etches these things in your long-term memory because they had some sort of heightened emotional element to it, because obviously those are important moments in your life that you want to remember. So that's dealing with the limbic system. It also activates your fight or flight system sometimes when it's a negative emotion. So anyway, there's a lot to do with the limbic system. Number 80, which ion gradient is primarily responsible for the resting membrane potential? So remember, resting was at -70. You got A, equal sodium and potassium concentrations, B, higher intracellular sodium than extracellular, C, higher intracellular potassium than extracellular with selective permeability to potassium, and then D, high intracellular chlorine. Well, let's look at any cell. Okay, we know that we have high amounts of sodium outside of it, got high amounts of potassium inside, and then chlorine is actually high outside typically, but the reason the cell is negative is because there's a bunch of proteins packed inside the cell that have this strong negative charge. Now, a lot of the times, in neurons especially, they will have these little leak channels that they typically call potassium leak channels. So what that means is that there's some permeability for potassium to leave, and if potassium leaves, we will make that voltage of the cell more negative, thus establishing our -70 millivolts resting membrane potential. So the best answer for that one would be, if I can find it, it would be C, high intracellular potassium with selective permeability, able to leave or enter to potassium. So by losing potassium, we maintain that resting potential. Awesome. Number 81. Action potentials are conducted faster in axons that, think about our power lines. What makes those electrical impulses fast? We've got A, are thinner and unmyelinated. B, have a large diameter and are myelinated. C, are shorter and lack nodes. Or D, lack sodium channels. Well, we know that thin wires can kind of get broken down and the electricity can get screwed up, so it's not A. B, they have a large diameter and are myelinated. Well, that means there's a insulation wrapped around the tube of the axon, so therefore that's going to speed up that conduction cycle, just like our power lines, very thick and insulated so the electricity can flow quickly. So it would be B. If it was D, lack sodium channels, no neuron would fire. We need those sodium channels to let the sodium in so that we can fire the neuron. Number 82 is kind of a know it or you don't. Which neurotransmitter is commonly excitatory in the central nervous system? We've got GABA, B, glycine, C, glutamate, or D, serotonin. Typically, glutamate is going to be a stimulatory neurotransmitter in the central nervous system, usually more in the spinal cord. A and B, GABA and glycine, are usually inhibitory, and serotonin can kind of be a neuromodulator depending on its target. So the best answer would be C for that one. 83. Which mechanism terminates neurotransmitter action in the synaptic cleft? We've got A, the degradation by enzymes, reuptake into presynaptic terminal and diffusion. We've got B, only diffusion away from the synapse. C, permanent binding to the receptors. Or D, endocytosis of the entire synapse. Okay, let's check that one out. So in the neuromuscular junction, as an example, here's our neuron, and then here's going to be our muscle cell here. We know the neuron will release acetylcholine, kind of has two components to it, into the synapse, and typically it will bind to those receptors to open up the gates. Now, when we need to turn it off, we, number one, need to cleave that down. How do we do that? We use an enzyme to break it apart into individual units. Now, from there, we need to reuptake it back into the neuron via diffusion or selective transport. So therefore, it's going to come back into the neuron and actually get repackaged as acetylcholine. So the best answer would be, what is it? I can't find it. 83. We are going to have degradation by the enzymes. We're going to have reuptake and diffusion. So the reuptake is kind of similar because we're diffusing it back across the membrane. So it would have been A. Awesome. 84. Myelination in peripheral nervous system is performed by, again, it's a know it or you don't. Anytime we're talking about the peripheral nervous system, know that the glial cells both start with the letter S. Schwann cells, satellite cells. So we only have one in here. It'd be the Schwann cells. That would be B for the correct answer. And they do myelinate those peripheral nervous system axons in sensory and motor neurons. 85. The blood calcium level is lowered primarily by, so remember we talked about this earlier, parathyroid hormone raised blood calcium. Equal and opposite will be calcitonin. So the correct answer would be B, calcitonin for this one. Number 86. Which anatomical term correctly pairs with its description? The tibia is the what? A, the thigh bone, B, the medial bone of the lower leg, C, the lateral bone of the forearm, or D, the bone of the ankle. Well, we know A, the thigh bone is the femur, that big bone in your thigh. B, the medial bone of the lower leg. Well, we've got two bones in the lower leg. We've got the tibia and we've got the fibula on the lateral side. Medial side would be the tibia, so it'd be B. The lateral bone of the forearm would actually be the ulna in anatomical, or sorry, I said that wrong. It would be the radius, anatomical position. It's on the lateral side. The ulna would be the medial side of the forearm. And then the bones of the ankle would be like the talus, navicular, all of those tarsal bones. Okay, so that'd be 86. 87. The pelvic, think about the pelvis, girdle is primarily formed by the what? A, the scapula and the clavicles. Well, that's up here, right? So we got the clavicle and the scapula here. B, the coxal hip bones and the sacrum. Hip bones and the sacrum, that's going to be your pelvic girdle because it's in your pelvis. Sternum and ribs are going to be right here, and then vertebrae only, all not related to the pelvic girdle. So that would have been B. Number 88, which structure transmit mechanical force from a muscle to a bone? So in other words, what connects a muscle to a bone? We've got ligaments, we've got periosteum, bursas, or tendons. We always know that bone to muscle is a tendon. Bone to bone is a ligament. Awesome. So that would have been what, D. Number 89, the slow oxidative muscle fibers are best for what? So slow oxidative, long-lasting aerobic respiration. So we've got A, short, powerful bursts of movement. Does that make sense? Probably not. B, we've got rapid eye movement only. So like darting your eyes back and forth, up and down, back and forth. Eventually your eyes kind of get tired. So probably not that. It's kind of more of a rapid burst. C is anaerobic sprinting. Anaerobic refers to without oxygen. So it would have to be D, none of the above. This would be much more like long walks or slow endurance distance running would use slow oxidative muscle fibers. Awesome. Number 90, we got 10 left. You guys are crushing it. In skeletal muscle, the term motor unit recruitment refers to what? We've got the number of sarcomeres that shorten, the sequential activation of more motor units to increase force. We've got the division of muscle fibers into separate muscles, or D, the refilling of ATP stores. Whenever we're talking about a motor unit recruitment, remember we were talking about basically one neuron per a certain number of muscle fibers. So if these muscle fibers are, say, in your bicep, and you're picking up a marker, you're not going to recruit very many motor units. But then if you need to pick up something heavier, like this entire whiteboard, you've got to recruit a lot more of those motor units. So if you think about the answers, it would probably be B, the sequential activation of more and more motor units, so more and more neurons with more and more muscle fibers that they're innervating, so that by using more muscles, we can generate more force. So that'd be the correct answer. Now, I want to mention that A would also be true. We're shortening more sarcomeres. That is absolutely true with this, but it's talking about the motor units, the neurons recruiting those muscle cells in general. Awesome. Y'all, I'm on to my last page. Also, can you read that print? I am very young. I can still read very tiny print. All right, let's move on to 91. Which is a primary function of connective tissue. I always think of connective tissue as cells, gels, and fibers. So what are they going to do? Are they going to conduct nerve impulses? Well, that's nervous tissue. Are they going to support and protect and bind other tissues? That sounds right. So we're going to bind things and kind of wrap things, protect things. That could be true. Production of action potentials, again, in nervous tissue. And then movement of body structures is all muscular tissue. So the correct answer would be B. 92, moving right along. Which of the following describes isotonic concentric contraction? Okay, so let me demonstrate it first so you know what it is. When we are picking up a weight, like with our arm, hopefully you can see it, when I'm contracting my bicep, what I'm doing is doing an isotonic, same tension, shortening of my muscles, concentric exercise. So I'm shortening my muscles under force. That's concentric isotonic exercise. If I hold it here, this is actually called isometric. Same length of muscle fibers, but my muscle's still contracted. So it's isometric. But then we can also lengthen my bicep under force. That would be isotonic, but it'd be eccentric. So which one would this be in this case? We're talking about isotonic concentric contraction. So we're talking about this part where we're going up. A, the muscle lengthens while generating force? No. B, the muscle generates force without changing length. Well, we're changing length here. C, the muscle shortens while generating force. That would be the correct answer. And then D, muscle relaxes actively. So that would be the correct answer, would be C. Now, B, muscle generates force without changing length. That would be isometric contraction. We're generating force, but we're not changing the length of the muscle. All right, moving on to 93, the perimysium, okay, wrapped around muscles, right? Peri around, mysium means muscle. What does it surround? A, individual muscle fibers. B, the fascicles or the bundles of muscle fibers. The entire muscle only. C, or D, the neuromuscular junction. Well, when you look at mesium, it's basically meaning the muscle wrapping. So we're going to be wrapping around something with the muscle. So it's not going to be the neuromuscular junction because that deals with neurons. Let's look at A, individual muscle fibers. Well, individual muscle fibers are individual cells, but if we look inside of a whole muscle, what we will see is basically a big old tube with a bunch of individual fascicles that contain muscle fibers. So if I were to draw it, it kind of looks like this. What we have, this is the whole muscle as itself. On the outside of the whole muscle, we have something called epimysium. Epimysium, okay, basically meaning upon the muscle. Then we will have endomysium all the way throughout, but then wrapped around the actual mesium, man, if I can spell right, mesium, but then wrapped around the fascicles themselves, so this is a fascicle containing all the muscle fibers, we will have a wrapping called, as we see, perimysium. So therefore, the correct answer would be it's wrapping around what? The muscle fascicles. So that would have been B. Fascicles are the bundles of muscle fibers. Moving on to number 94. Which ion channel opening would hyperpolarize a neuron? So anytime we're hyperpolarizing a neuron, we've got a negative resting potential, negative 70. But then hyperpolarization means we're going down. So we're getting more negative. So which one would make it more negative? We've got A, voltage-gated sodium channels opening. Well, sodium would flow into the cell in that case, so it would make it more positive. We've got B, calcium channels opening. Well, again, calcium flows in and it's positive, so that'd make it more positive. C, potassium channels closing. Well, in that case, potassium is neither leaving nor going in, so nothing's happening there. And then D, chlorine channels opening, allowing an influx of calcium. So in that case, if we have a cell and now chlorine as a negative charge is going to go into the cell, it will make it more negative. So that's absolutely true. It would have been D, chlorine channels opening, allowing chlorine to come in. All right, moving on, 95. Which receptor type on sensory neurons transduces chemical stimuli? Chemical, the prefix is chemo. So if you look at all those answers, we've got mechano, chemo, photo, thermo. The correct answer would be B, chemoreceptors. Mechanoreceptors typically detect force, photoreceptors detect light, and then D, the thermoreceptors detect temperature. So if we were asking a question about those things, those would be the receptors that we are using. Okay, y'all, we have 5 more. If you are still here, I'm going to help you out a little bit for that free organized biology swag. If you comment physiology rocks, I will put your name in twice for that drawing, okay? So put in the comments physiology rocks and you have a higher likelihood of getting one of these free shirts or a mug or whatever else that you want. All right, moving on. So 96 here. The primary structural difference between cardiac and skeletal muscle is what? We've got A, cardiac muscle cells are branched, interconnected, and have intercalated discs. Those are all true things, so maybe that's going to be the right answer. B, cardiac muscle is voluntary, skeletal is not. Well, you know cardiac muscle is involuntary, right? If it was voluntary, I would forget about it constantly. So it's not that one. C, skeletal muscle has gap junctions like cardiac muscle. Now, the gap junctions are only found in cardiac muscle by definition. And then D, cardiac muscle lacks sarcomeres. Well, we know both cardiac and skeletal have sarcomeres. That's what makes them striated. So that would be incorrect. So the correct answer would be A. Moving on. Which statement about epithelial tissue is true? We've got A, epithelia, highly vascularized with many blood vessels. B, epithelia have abundant extracellular matrix. C, epithelial seldom form glands. Or D, epithelial also always rest on and are supported by connective tissue. Epithelium does not have any blood supply. So anytime we're talking about like epithelial or lining cells, they are completely dependent on the blood supply below them from the connective tissue. So here's connective tissue, here's the epithelial tissue. So it gets the blood supply from the connective tissue. It doesn't have it in and of itself. So it would not be A. B is it has abundant extracellular matrix. Well, we know with the matrix, it's primarily connective tissue where you have like your cells, your fibers, and some fluids in the extracellular matrix. So the epithelial tissue doesn't really have that. C is epithelia seldom form glands. Well, the epithelial tissue are basically the glands. We can have endocrine glands that secrete things in the bloodstream, exocrine glands that can secrete things out of the body. So epithelial tissue is basically the only tissue that forms glands. So the only correct answer could be D. Epithelial always rest on and are supported by connective tissue. That is absolutely true. They usually call this the basement membrane. So the basement membrane here would be connected to all the connective tissue underlying it. So you may get that question on a test as well. 3 more. In bone growth, appositional growth means growth, A, in length at epiphyseal plates, B, in width by addition of new bone to the surface, C, by replacement of cartilage only during embryogenesis, or D, only after fractures. In this case, appositional basically means like opposing a position. So in this case, typically bone will lengthen. So here's like a bone, and typically we think of bone growth as lengthening. But then appositional growth would basically be the opposite. We're going opposite direction. So this would be appositional growth, where are we depositing bone on the surface and making the bone thicker. So in this case, it would be what? By width, B, in addition of new bone to the surface. That's appositional growth. 99. The primary function of myoglobin in muscle cells is to what? Now, if you look at the word myoglobin, myo always refers to muscle. Globin always refers to a carrier protein of some sort that's holding on to something. Okay, and we'll see what it is. A, to break down ATP. Well, ATP is always broken down by enzymes called ATPases, so that couldn't be correct. B is store oxygen for use during contraction. Well, maybe we could use some oxygen. C is transport ions across the membrane. Well, maybe it's carrier protein, so maybe it's carrying something. Or D, catalyze glycolysis. Well, glycolysis is catalyzed by other enzymes, so it wouldn't be that one. Well, we know that myoglobin is incredibly similar to a protein called hemoglobin. Hemoglobin and myoglobin both will bind oxygen, helping us do aerobic cell respiration. So the correct answer would be B. It stores oxygen for use during contraction. Transporting ions across the membrane for C would typically be something called a channel protein. So like here we have a cell and we have some sort of channel protein that's allowing ions or something to come through. That's very different from myoglobin. It's a really big protein that holds onto oxygen for the skeletal muscle. Last one, and then I'm going to send you on your way. Number 100, which process most directly increases surface area for absorption in the small intestine? We already talked about this one. We know that it is these columnar cells that will have those microvilli, but we also know that all the columnar cells are organized in this fashion throughout the small intestine. So if you look, they're lining things like this. So these big, long finger-like projections are called villi, whereas the cells themselves will have these microvilli to increase the surface area. So the correct answer would be C. Best of luck. Can I pray for you real quick? Lord Jesus, we thank you for the opportunity to study the human body, and I pray that you would bless whoever is watching this with wisdom, with knowledge, with focus, so that they can ace their test, be successful, and then go on and be a great healthcare provider, blessing other people as you have so greatly blessed us in Christ. So Lord, forgive us, empower us, and help us to do all these things in your name. Amen.